Hi, it's Dr. Rosario, and in this video we are going to continue our discussion of viruses. Today we are going to talk about viral replication cycles. We will focus our attention specifically on animal viruses and how they get into the host cell, take over the cellular machinery, and make copies of themselves. I will talk about host specificity, viral genomes, and how viruses cause damage to the host. It is important to remember that there are viruses that infect every type of cell. Pictured on the left are viruses that are invading an E. coli cell. In the middle is a tomato that has been infected with spotted wilt virus. Finally, on the right we have HIV, the human immunodeficiency virus. It's seen there in green emerging from a human cell. While that tomato doesn't look quite right, I can eat it without having to worry about catching spotted wilt disease from it. That is due to host specificity. Host specificity describes what type of cells a virus can infect. It is not just limited to the type of organism, but also the type of cells or tissues within an organism that can be infected. For example, HIV only infects the cells of the immune system. This type of host cell specificity is called viral tropism. A viral preference for one type of cell over another depends on how the molecules on the surface of the virus interact with the molecules on the surface of a potential host cell. There has to be molecular complementarity. The two molecules must have a complementary shape and be able to form non-covalent interactions. Host range describes the variety of cells that can be infected by a virus. For example, a virus that has a restricted host range would be something like hepatitis B, that only infects the cells of the human liver. A virus with a moderately restrictive host range would be poliovirus, which infects intestinal and nerve cells of primates. A broad host range would be something like rabies. Rabies infects various cells of almost all mammals. Viruses need a host cell to make copies of themselves so they can spread to new hosts. They take over the cellular machinery for the purpose of making as many copies as possible. This process occurs in a series of steps that repeats with each new host cell. We will focus specifically on how this occurs in animal cells. The stages of viral replication are adsorption, penetration, uncoating, synthesis, assembly, and finally release. We will talk about each one of these in detail. The word adsorption is used to describe when a molecule sticks to a surface. As I described when talking about host specificity, viruses stick to host cells through interactions between viral surface molecules and the molecules on the surface of the host cell. These interactions are highly specific and depend on molecular complementarity. Once a virus sticks to a cell surface, it can then enter or penetrate the cell. Viral penetration can occur through endocytosis or via membrane fusion. Here I illustrate the process of receptor-mediated endocytosis. This virus binds to the host cell using the spike proteins that are part of its capsid. These proteins interact with the specific receptors on the host cell surface. Once it is bound, the cell begins to form a pocket in the plasma membrane. Eventually, the membrane pocket will completely close off to form an endocytic vesicle, or endosome, within the cytoplasm of the cell. Another way that a virus can penetrate a cell is via membrane fusion. This can only occur with enveloped viruses. Here, the spike proteins are embedded in the viral envelope and bind to the host cell receptors. Instead of a pocket forming within the membrane, the viral surface is brought closer to the host cell membrane, and the lipids of the envelope fuse with those of the plasma membrane. The viral envelope is left behind, and the nucleocapsid is released into the cytoplasm of the cell. Once the virus enters the host cell, the next step is to release the viral DNA or RNA into the cytoplasm. The envelope and the capsid are not needed inside the cell. For viruses that enter through endocytosis, uncoating of the viral nucleic acid happens inside the endosome. Changes in pH cause the capsomeres to disassociate from one another, and the capsid falls apart, releasing the viral nucleic acid. This is then transported to the cytoplasm. For viruses that enter via membrane fusion, the process is slightly different, but the end result is the same. The capsid falls apart, and the viral nucleic acid is released into the cytoplasm. Once the viral nucleic acid enters the cytoplasm, the next step is synthesis. Here, the cell will use the information stored in the viral genome both to make copies of the genome and to make new viral proteins. In cells, the flow of genetic information is usually from DNA to RNA to protein. Viral genomes, however, are not always composed of DNA, and this makes the viral synthesis process more complicated. Viral genomes can be composed of double-stranded DNA, single-stranded DNA, plus sense single-stranded RNA, negative sense single-stranded RNA, double-stranded RNA, or we have retroviruses that have plus sense single-stranded RNA but fit into their own special category. For a cell to read the information stored in each of these genomes, different steps have to occur. Let's look at a double-stranded DNA genome first. Here, the viral genome is most similar to a cell's genome and can be read in the same way. Once the viral DNA is inside the cell, it is usually transported into the nucleus, where it can be used to make mRNA, which then can be used to make proteins. The cell's DNA replication machinery can be used to make copies of the viral genome. Together, the newly made viral proteins and DNA can be assembled into a new viral particle. Single-stranded DNA viruses are a little different because the single-stranded DNA must first be copied into double-stranded DNA before the process can continue. Once the double-stranded DNA is made, it can be used as a template for mRNA synthesis and production of new viral genomes. Let's take a look at a typical DNA virus replication cycle. In this example, I'm going to use an adenovirus, which is often the cause of the common cold. Adenoviruses are naked icosahedral viruses with a single linear piece of double-stranded DNA as its genome. The first step of the life cycle is adsorption, where the proteins on the surface of the virus bind to host cell receptors. This virus enters the cell via endocytosis. This is the penetration step. Inside the endosome, that vesicle that is formed during endocytosis becomes acidified. The drop in pH causes the viral capsid to disassemble, leading to uncoating of the viral DNA. The DNA escapes the endosome and is transported into the nucleus of the host cell through nuclear pores. For most DNA viruses, the synthesis of new virus begins here in the nucleus. The DNA can be read by the host cell's machinery to make mRNA, which is pictured here in green. That mRNA can in turn be used to make viral proteins. The viral DNA will also be replicated to make new copies of the viral genome. Assembly of the completed virus occurs in the cytoplasm, and this particular virus causes a cell to break open or lyse, releasing the virus to infect new host cells. Now we will turn our attention to RNA viruses. Once the genome of a plus-sense RNA virus enters the cytoplasm of the cell, it can be read immediately to make proteins. Some of these proteins can be used to form the structure of the new virus, while others can be used to replicate the viral genome. The plus-sense viral RNA is copied into a complementary negative-sense RNA template that can be used to make many copies of the original genome. Together with the structural proteins, these can be assembled into new viruses. Negative-sense RNA viruses are a little different because their genomes must first be copied into plus-sense RNA before they can be read. That new RNA can be translated into protein or used to make new negative-sense RNA genomes. For a double-stranded RNA virus, the viral RNA is used to make plus-sense RNA that can be used to make viral proteins and the new double-stranded RNA genomes. So here we have an example of an RNA virus that we are all familiar with, SARS-CoV-2, the virus that causes COVID-19. This is an enveloped plus-sense RNA virus. The virus first attaches to host cells of the respiratory tract. Adsorption occurs through interactions between the protein spikes found in the viral envelope and cell surface receptors called angiotensin converting enzyme 2, or ACE2. The viral envelope fuses with the cell membrane, and the helical nucleocapsid is released into the cytoplasm. The capsid proteins dissociate from the plus-sense RNA during uncoating. This RNA can be immediately read to make proteins or can be copied into a negative-sense RNA template to make viral genomes. The viral proteins can be made directly in the cytoplasm, like the capsid proteins, or they may enter the secretory pathway, traveling through the endoplasmic reticulum and the Golgi, like we see here with the spike proteins. In the case of SARS-CoV-2, a specialized vesicle is formed, and the newly assembled nucleocapsid buds into the vesicle to form the envelope. The completed virus then leaves the cell via exocytosis. Retroviruses are a unique group of viruses. These viruses are RNA viruses in which the genome is reverse transcribed into DNA within the host cell. The viral genome can then be read by the cell's machinery like any other DNA to make mRNA and proteins or to produce new viral RNA genomes. The human immunodeficiency virus, HIV, is a retrovirus. This is an enveloped virus that binds to a cell surface receptor called CD4 that is found on some of the cells of the immune system. CD4, along with a second surface protein, interacts with a viral spike protein, which leads to fusion of the viral envelope. The nucleocapsid, containing the viral RNA along with some essential viral proteins, is then delivered into the host cell cytoplasm. Once inside, the capsid releases its contents And the first viral protein, reverse transcriptase, pictured here in red, uses the viral RNA as a template to make new double-stranded DNA molecules. Integrase, pictured here in green, then helps transport the new DNA into the nucleus of the cell, where it inserts the viral DNA into the host genome. The virus can remain within the host genome indefinitely without being read. It is not entirely understood what events trigger transcription of the viral genes. But that then produces new viral mRNA and viral proteins that can be assembled into new viruses that bud out of the surface of the host cell. The HIV virion is not complete until the final viral protein, called a protease, pictured in purple in this figure, activates the capsid proteins, allowing them to assemble into the completed virion that then can infect the next host cell. As we have seen with the few examples of viruses that we've discussed in this video, assembly can occur within different cell compartments, including the cytoplasm, the nucleus, or the endomembrane system. Viruses can be released from host cells via budding, like we saw with HIV, pictured here, exocytosis, which we saw with SARS-CoV-2, or by causing the cells to burst or lyse. Viruses can cause a wide array of damage to cells. That damage can be direct or indirect. Viral infections can lead to cell death through cytolysis, apoptosis, or necrosis. Cells can become disoriented, undergo major changes in shape or size, or develop intracellular damage. These changes in the microscopic appearance of cells are called cytopathic effects. An example of some of the damage that we can see are inclusion bodies, which are compacted masses of viruses or damaged cell organelles in the nucleus or cytoplasm of a cell. The dark staining granules in the cells pictured on the left side of the screen are an example of inclusion bodies that have formed in cells infected with a human herpes virus. Another example of a cytopathic effect is the formation of syncytia. Syncytia occur when multiple host cells fuse together into a single large cell called a giant cell that contains multiple nuclei. This is pictured on the right-hand side of your screen. Here you have 6 cells that have fused together to form a giant cell in response to a measles virus infection. Sometimes the damage from a viral infection is indirect. The host immune response to a viral infection can cause significant damage to tissues, for example. In these images, you can see 2 lung x-rays. One is of a normal, healthy lung. The other is of a patient with COVID-19 pneumonia. That's the one on the right. As a result of the SARS-CoV-2 infection, inflammatory chemicals are released by the body that can damage the lungs and cause them to fill up with fluid. This can result in the most severe forms of COVID-19, in which patients require mechanical ventilation in order to survive, and the lungs can be permanently damaged. We don't fully understand yet why this happens in some cases, but not others. In many cases, viral infections can lead to damaging inflammation. Some viral infections can alter cell behavior, leading to the development of cancer. These are known as oncogenic viruses. It is thought that up to 20% of human cancers are caused by viruses. Some known oncogenic viruses are hepatitis B virus, hepatitis C virus, human papillomavirus, Epstein-Barr virus, human herpes virus 8, and HIV. Most commonly, DNA viruses and retroviruses are associated with cancer. Cancer is uncontrolled cell growth. There are several ways that viruses can cause cancer. Proteins that are encoded in the viral genome may disrupt the host cell cycle control system. Integration of viral DNA into the host genome can impair normal genome function. And chronic inflammation from persistent viral infections can also lead to cancer. Most viruses are eliminated from the body after a short infection period. Some viruses, however, can persist in the body for years, establishing a lifelong infection. Some of these can become latent after an initial period of illness. Latency is when a virus is present within a host but is not actively replicating. Sometimes the virus can even be incorporated into the host's DNA as a provirus. An example of a persistent viral infection is the virus that causes chickenpox. Upon initially entering the body, varicella zoster virus replicates rapidly, causing fever and a characteristic itchy, blistering rash pictured on the left side of your screen. After about 2 weeks, the rash goes away and the patient appears to recover. The virus, however, continues to persist in nerve cells without replicating. In some cases, if a person becomes immunocompromised, the virus can start to replicate again and cause a painful localized rash called shingles, which is pictured on the right. It's not fully understood why some people develop shingles and others don't, but we do have a vaccine that can protect against it. There are hundreds of viruses known to cause diseases in humans. There are likely many more yet to be discovered. Understanding how these obligate intracellular parasites reproduce is key to learning how to stop these diseases that they cause.